The group is constituted by basic and translational researchers and also for clinical researchers. For us, it's very important to be involved, to involve clinicians in our research and working together for better understanding the limitations and the gaps that clinician has for managing the patients. Then in the clinical research, we can see internists, intensivists, rheumatologists, pulmonologists, preventive medicine, pediatrician, and pulmonologists. And on the other hand, we have two research line leaders, that is Irene and Alicia, postdoc researchers, master students, PhD students, from the Odessa University, in fact, and the support of management and tech transfer units from the institution. Our group is working in five research lines. One is the microbial interaction. We are thinking here in biofilms, the immunoresponse characterization, how the host is responding against the different microorganisms. We have a line that is focusing, evaluating the impact of the external factors in the balance among the immunity and the pathogens, because external factors like pollution, plastics, tobacco, diesel, it could attenuate the immunity. Then we have working in diagnostic technology innovation that is trying to develop very new methods for improving the diagnostic. And we have also working in new therapeutic approaches. And we are working in bacteriophage, we are working in new compounds, some of them from Black Sea, we are working in antivirulence drugs, we are working also in gallium, as an example. We are working also with chemistry with the intention to include these compounds in nanoparticles to facilitate that the drug reach the place where the bacteria or the microorganism is. And as you can see, the group is distributed, the members of the groups are distributed in the different research lines, including the researchers from the Odessa University. As mentioned in my presentation, we are leading an Innova for TB consortium. It's an European project that involves 16 institutions from 6 different countries. One of them is Ukraine, and Odessa University is one of the most important partners in the consortium. And the consortium is constituted by universities, it's constituted by research institutes, hospitals, but also companies. Which is the idea? The idea is that in the knowledge and the skills from the academic institutions, hospital, and Smith was exchanged, and we exchange staff among these kind of institutions. Then the Innova is based in the secondment of participants in an international way and in intersectoral way. And here also you can see the different work package, that scientific work package of the Innova, that is the host pathogen interaction, development of new diagnostic methods, development of multi-drug resistant TB detection tests, and also the improvement of therapy strategies. We are really proud of our training program. Our training program is based in intersectoral and international exchange and secondment visits from a staff of one institution to another institution. It is possible to perform secondments, early stage researchers or experienced researchers, but it's also open for technicians and managers. When we mention early stage researcher, we are thinking in young PhD doctors or PhD students. The secondment, it could be from 1 month to 1 year. Currently, for Ukrainian participants, it could be even longer. For each Participant, we assign an international mentor and international supervisor with the idea to guide not only during the secondment in the research activity, if not also during the research career, that it could be longer. And the idea is to learn about science, but also transversal and specific skills, that it could be to perform presentations in English, or it could be participating in Congress, preparing abstracts or posters, or even writing manuscripts. Now we are really happy because we have received the news that a new project has been granted by the European Commission. It's a cost action. We received the news at the end of May. The new project is called Advanced TB, and it's including all the Innova4TB participants, but we have increased the family. And currently we are involved 38 institutions from 22 countries. And it's increasing because we are receiving applications from all the participants that will be involved in the network. You can see a small figure, the number of the countries that are involved, and also Guatemala, Chile, and Nigeria. The objective of the new project is promote the networking and promote the training of the participants. Then we have different working groups focusing on clinical cohort of patients and dissemination and therapeutics and treatment. And the idea is to promote the summer schools, promote exchange, promote activities and meetings for learning and increasing the skills of the participants. This is a very good news for all the consortium. Now, go in deep in the topic of the lecture, that is the tuberculosis and the COVID, how it's coming in the future. Then first of all, it's important to mention that there is some similarities between tuberculosis and COVID, because both are respiratory diseases, both are air transmission with similar preventive measures, distance, face mask. We know the hospital infection control to avoid the nosocomial infection, and it's the same contact tracing strategy that we follow for know who is in risk to be infected after exposure with a patient. Then we have also the microbiology and the immunology knowledge. We have networks of clinical trials, and we have in our laboratories and other facilities and capacities for working with both microorganisms. For this reason, we have created an Innova4COVID consortium, very similar to the Innova4TB, but focusing on COVID, that we are running in parallel. Then it's a consortium that is involving companies and institutions across Europe, where we have been involved in different clinical characterization studies. And also we have developed a diagnostic platform for evaluating new technology and assessing the immune response against COVID. And we are involved also in vaccine clinical trial. One of them was to study the BCG vaccination. BCG is the vaccine of the tuberculosis. It has some impact in reducing the severity of the COVID in healthcare workers. And we have also studies evaluating the immunogenicity against the vaccines in older individuals. Tuberculosis, for sure, that it represents a major problem. This is the data of the last year. Almost 10 million of people fell ill, with 1.5 million died. A quarter of the population considered that it's infected. This is a high amount of patients that are severely ill, and a high amount of patients that is dying every year, because this is the number of patients for 1 year, but you should multiply this for all the years that the tuberculosis is in the world. COVID is also a serious disease that started recently, in fact, in December 2019 in Wuhan, in China, that it spread very quickly and very fast around the world, provoking an incredible pandemic that with a high mortality at the beginning, because we have not immunity and no vaccines. And currently, this amount of confirmed case has been diagnosed until yesterday, with more than 6 million people dead in a couple of years, but with a high amount of vaccines administered at this moment. This has been lucky because the vaccination has helped to reduce the impact of the disease. But until now, the COVID pandemic has had a major impact on the capacity of the health systems, has induced problems in reproductive health, has impact in non-communicable diseases, in routine immunization, in maternal and child health, in malaria also, in HIV control, have increased the poverty, but also has impact in the tuberculosis control. Then the major impact of the COVID has been because it has disrupted the systems for controlling and managing the tuberculosis. It has been a problem for following the treatments and preventing the TB treatment for infected individuals. And in the last year, it was described that TB deaths increased for the first time in over a decade. Few people were diagnosed, not because we have less patients, if not because we have not made the same effort for diagnosing the TB patients, and the treatment, it also was decreasing. There was a fewer resources for essential TB services and research in TB. Regarding diagnostics, the methods for TB, as you probably know, is the smear examination, the culture, that is slow, and some molecular methods. For COVID diagnostics, we have not a smear because it's a virus and we need other technology, but we have developed very quickly antigen detection and also different PCR tests for detecting the virus. It's important also to comment the clinical spectrum, the immunospectrum that we can find in tuberculosis and in SARS-CoV-2. In tuberculosis, there is a spectrum of situations of patients with TB that is going from patients that are exposed but the disease is eliminated and you are not infected because your immune response, your innate immune response or your acquired immune response is able to control and kill all the bacilli. The latent infection, that is where the bacteria is in a latent phase inside of your body. You are not ill and you are not contagious, but you are infected and you could progress in some moment. And then the subclinical TB disease, that is when the disease that was in a latent stage start to progress to an active form, and the active TB disease, that is when the bacilli is multiplying in the lungs or in the tissue and you are ill, and then you are symptoms, and then you could contagious. In fact, in subclinical TB, you could also be contagious. The same happens with the COVID. The COVID has a degree in a spectrum of forms, because we can move from individuals that was never exposed to the COVID, individuals exposed without symptoms, individuals that exposed infected but without symptoms, with mild symptoms, really severe symptoms, and then patients that recover from this disease, patients that recover from a mild situation, from a mild disease, or from a severe disease. But the situation, it could be even worse or more complicated, because in the case of the TB, we can see also patients that were treated in the past, and now they are healed. And in the COVID, all this group of patients could be vaccinated, or it could be first infected and then vaccinated, or vaccinated first and disinfected, or even patients that are suffering a long COVID or have sequela in the lung as a consequence of the disease. Then the spectrum of clinical situations is high, and the immune response in each of case, it could be also different. There is also some interaction between the TB and the COVID. The TB patients has lung damage, has alterations in the architecture and the parenchyma of the lung, and this it could if you are then infected by COVID, it could increase the damage of the lung. And the TB generates some kind of immunosuppression, that it could be protecting against SARS, because it protects against the storm of cytokines. Then this balance is very interesting to study. And for doing this, we need to characterize as much as we can the immune response against both microorganisms. Talking about tuberculosis, we start exploring the immunity several years ago. In 2008, we published our first article evaluating the T-cell response that produce interferon gamma in different group of patients. Is that the technology that we know, like ELISpot or quantiferon, and we can see in that moment that using specific antigens like HRCs or CFP10, it's possible to identify better than with the tuberculin skin test the individuals that are truly infected in comparison with vaccinated alone or with NTMs. This is the ELISpot, the reading of the ELISpot. You can see the bottom of the well, where there is a membrane where the T cells that are responding to the antigens produce a dot, are producing interferon gamma that is detected in an immunoassay. The negative controls, the positive controls that are stimulated with a phytomagglutinin, and the response against the specific antigens. Then you can see here a negative result because the number is under the cutoff, and a positive result where the spots are clearly visible. This is a very good technology that it could help a lot for identify the individuals that are infected. It's unable to distinguish among infected or active, but it could be very useful for identify the patients infected and taking therapeutic decisions. Given that is enough for a better characterization, because we are only seeing the T cell and the interferon gamma, we have with Irene La Torre different studies doing exploring the immunoresponse. And then we have published and others, and we have seen how the immunoresponse is really, really diverse against the bacteria, and it depends of the antigen that you are exploring, the different subtype of T cells that is stimulated, and it's producing a different type of cytokine. And this, it could be also different depending if you are a child, a teenager, or adult. And this is a good explanation why it's very difficult to develop a vaccine that was useful against the pulmonary tuberculosis in adults, because the response is really, really complex. In the case of the SARS-CoV-2 and the COVID, we know that the lesions, the infection of the virus generate lesions in the alveoli, and it produces an acute respiratory distress syndrome with affectation of the parenchyma that is exacerbated by a cytokine storm. We have seen that in the SARS-CoV-2 it's possible to detect antibodies, IgG and IgM against the virus. Here we have a control group of individuals theoretically not exposed, where the majority of cases are negative, and only a few percent of them were positive or weak positive, that it could be cross-reaction with past coronavirus infection or not well-documented exposure. But we can see that individuals with a COVID in the acute phase or in the convalescence are able to produce a high amount, especially in the convalescence after 3 weeks, a high amount of protecting antibodies. Regarding the T cells, different studies has been done for exploring the role of the T cells in managing the COVID patients. And it has been seen that the patients with severity, but also patients with mild disease, produce a high amount of T cells, especially in the convalescence phase. No, it's not the same in the acute phase. In the acute phase, it has been seen that this not produce a lot T cell response, but it is possible to detect in the convalescent phase. We have been described, it has been also described that patients with active COVID produce some kind of lymphopenia, a decrease in the number of lymphocytes that are protecting the individual and are producing the T cell response. And this is shown here, how healthy individuals have this amount of lymphocytes, T cells, but individuals with COVID that are going to intensive care unit or non-intensive care unit has a different profile. With the patients with a severity disease has a lower number of lymphocytes. And you can see here, depending on the severity of the disease, how the amount of lymphocyte is decreasing. Then the amount of lymphocyte in blood, it could be a marker of severity and even of risk of death. And it also explains how the amount of the T cell response, it could be decreased in the severe patients. This is a study performed in IGTP, not for our group, that they observe, they follow some patients, and a low number of patients that were infected with COVID, and they obtain a sample 4 months later of the primary infection. They receive a vaccine, and 1 year later, or 10 months later, they obtain another sample. Then we have samples after the primary infection and samples and after the primary infection and the vaccine. And you can see here in the patients that are able to react with producing antibodies that the profile of T cells is not the same, it's different. Then it means that the response against the COVID is also different. And it could be different if you are vaccinated or if you are infected, or you are infected and then vaccinated. The pool of antigens used and the exposure to the real virus stimulate the response to different T cells along the time. Now I'm going to explain our experience in COVID. We have performed studies of T cell immune response against the COVID that the T cells producing interferon gamma. And we catch the PBMCs from the blood and stimulate with the specific antigens of the SARS-CoV-2. And if the individual is infected or has a disease or is convalescent, and will produce, as a consequence of the stimulation, the interferon gamma, that it will be detected by ELISpot. Then for this study, we have included patients vaccinated and not vaccinated, patients with active TB, and patients after the disease in a convalescent stage. And we detect the antibodies that they are produced in all the groups. And we have collected the PBMCs and have stimulated with the antigens for detecting the T cell response. This is the summary of our data. It's a table that very briefly include the data against the different antigens, spike, nucleocapsid, and membrane in the different group of individuals. And it's very easy to see, if we focus in a spike, that the amount of individuals that are producing T-cell response in vaccinated is only 36%. And in non-vaccinated is almost zero, because theoretically they are not exposed. In acute phase, we can see how the amount of percentage is increasing with the severity, and how the amount of percentage of T-cell response is also increasing in the convalescent phase according with the severity of the case. And the same happens for nucleocapsid and for membrane, although you can see that the immunogenicity of the spike is higher than the nucleocapsid for almost all situations, especially in the severe where the percentage are higher. And obviously in vaccinated, the percentage is almost zero because nucleocapsid and membrane are not present in the vaccine. We can show this in figures, observing the amount of T cells that are produced. And you can see here for the spike that no vaccinated almost are zero or under the cutoff. For vaccinated, a large number of patients are not responding, but a high number of them see. Yeah. Then in the convalescence, a lot of response after the disease, increasing the signal with the severity, and also for the acute phase, increasing a bit in the moderate, not in the severe. That is something that it was described. The T cells is clearly present in the convalescence, but not in the acute phase, because has not enough time for creating this, for mounting this defense. The same for nucleocapsid, with no response in vaccinated. It's not so clear, but higher response in convalescence than in acute. And this is a different way to present the data. Here you can see the T cells that are producing interferon gamma in individuals vaccinated, according with the days after the vaccination. Then it's not perfectly clear, but at the beginning, with only one doses, there is no response. And then when we include the second doses of vaccination, it starts produce T cell response, not in all cases, but in some of them. And probably this justified to a third shoot of vaccine for stimulate finally all the T cells response again, at least these that are producing interferon gamma. And here is the response after the diagnosis in the convalescent phase. And it's for spike, for nucleocapsid, and for membrane. It's shown how the response increased in the majority of cases with the time. This is a comparison among the T cell response and the interferon, and the IgG, the antibodies. And we can see in this figure that the antibodies in black are produced almost in all cases, in a high number of patients, in comparison with the T cells response. Then this could show that this is the same. This is a high production of antibodies, although the number of T cells producing interferon gamma was low. And this justifies that probably antibodies is produced quickly and protect initially the individual, but then the T cell response is coming lower, but it's behind. And then at the end, you could have both of them protecting you until some way. Then, which is the future challenge that we have to face in the future for fighting against the tuberculosis also, but against the COVID? Or, okay, on one hand, from a view, we need work from now for preventing next pandemic, next virus coming from the wildlife. We know that more than 2 million of not well characterized viruses are in the wildlife, in other mammalians. And then there is a risk of changing of a species and start a new pandemic that it could be even more severe than now. Then this requires to work in teams, work in a multidisciplinary point of view, in multidisciplinary teams, and work in one world, one health, including the same research teams the humans, the animals, and the environment, because we are in the same place, all is connected. Then we need to involve biologists, ecologists, people that working with health science, engineering, social societies, human medicine, comparative medicines, and for sure, veterinary medicine. We need to work together for understanding better the way that follows on microorganisms for passing from the animals to the humans, and not pass the border for avoiding new pandemic. We have also learned, and we have also to apply to the tuberculosis, the Swiss cheese model. The Swiss cheese model is a very graphic, a very nice way to see how we should fight against a disease, in this case, the TB, but it's also useful for COVID. It means that all the actions, all the initiatives, all the methodology that we use is not perfect. It's a slice of cheese. It's not perfect. It protects in some way, but has some holes that it not avoid all the cases, it not solve all the problems. Then if we use the BCG, then the BCG is protecting a lot, but it's not enough. If we use face mask, it's protecting, but not enough. And the same for the technology. But if you combine different strategies at the same time and together, probably we will be able to stop covering the holes that one strategy has and the other have solved. We have to also consider in this graph the misinformation mouse, the misinformation, the fake news that are generating dupes and new holes that were not controlled from the beginning. And this is another problem that we have to control, that we have to solve for the future. Give real information, good information, and avoid or fight against the fake news that is affecting the control of the diseases. Dr. Morten Ruwel from the FINE described some time ago, 1 year ago, that we have to learn from the COVID to reimagine the tuberculosis diagnosis. We have developed an incredible amount of new technology for COVID in a very few, in a very few time. We have been able to develop digital tools, new sample collections, auto-diagnosis, artificial intelligence, multi-disease molecular technologies, real-time for data. This is very important to maintain in the system and to use for tuberculosis. Obviously, this was able to produce because it was invest a lot of money. But now that the money has been invested, we need to translate this information and this new technology for tuberculosis, because it's a terrible disease that is producing a high number of deaths every year. And this is related with the inequities. We should reduce inequities. COVID pandemic is affecting all the world, but because it's affecting all the world, it's affecting also the North countries. And this has the reason that we have invested a lot. Then what happens with the South countries or Eastern countries that have not receiving the same number of investment or the same number of vaccine the COVID-19 vaccines? This is something that we have to address to reduce inequity and to fight in the same with the same tools and at the same level in all places of the country. And also we need to adapt our strategy, how we manage the diseases. This is the case of Spain, where you can see the different waves of COVID that we have suffered in the last 2 years. And this is the last one. Well, the last one is maybe that days, but in winter we have an incredible peak of cases of the Omicron variant. But if we look in detail, this variant, it was not the variant responsible of the higher hospitalization, or it was responsible only on a few number of intensive care unit admittance, and it was not the higher responsible of deaths, then we have adapted our managing protocols for fighting to the disease in a dynamic way. Yes, at that moment, with the new variants that are coming, the response should be proportional to the amount of disease that is producing. Then we should focus in modify the strategy in primary and hospital care attention to move to focus in vulnerable population and high-risk population. We have to move from a zero COVID in general population to a zero COVID in the vulnerable, in the risk population. And then as a final challenge, we should push the government and the organizations and the institutions to invest in science, to invest in research, to invest in diagnostic methods, to invest in reduce inequities, and to maintain and reinforce the global health system. If we have done an effort to increase the number of technicians, nurses, and researchers, they should remain in the system, or even increase to avoid new pandemic and to reduce the burden of the disease, like tuberculosis that we still have. Then as a conclusion and for finalizing, COVID has impacted in the health systems, affecting a lot of diseases and a lot of systems, but especially in the control of TB. The immunoresponse is important to be studied. To assess the cellular response against tuberculosis and SARS is of utility in assessing exposition, progression, and also protection. Probably several challenges we have ahead, and we have to fight a lot for reaching. And from my view, is mandatory a good communication. We need to work as only one team between all the professionals involved in TB and COVID control and other diseases that probably will come in the future. And that's all. Thank you very much. You can see here the two Twitter accounts where you can see our activity. On one hand, the One Health Lab group, that is the name, the casual name of our team in the GTP, but also the Twitter of the Innova for TB. Well, it's true that we have Twitter, but we have also Instagram, Facebook, LinkedIn accounts that you could follow us in the different social networks. And you can see here the webs, the official webs, and my email and the email of the consortium, just in case there is a mistake. Okay, just in case you want to contact us or learn more about our activity. And yeah, now thank you very much again. And I stop the presentation, and I will be extremely happy to answer your questions.
Okay, thank you very much, Dr. Dominguez. Are our participants have some questions? Okay, I have one. Dr. Jose, could you answer? We know that, as you highlight, that BCG vaccine reduce the severity of the COVID cases. Are the mechanisms of this known for now?
Yeah, it has been observed. The study is still not closed, but it has been observed that the BCG is not avoiding the infection, but is protecting for the severity. And this it was seen in the past for other diseases. It has been seen how in studies performed in Africa at the beginning of the BCG vaccination use, how children vaccinated with BCG reduce mortality, not only against COVID, not only against tuberculosis, if not against other diseases, other pneumonias, other viruses, including influenza. And this has been described as a training immunity. it has been observed how the BCG, it could modify epigenetically the DNA of the host at level of the inflammatory cytokines and inflammatory markers. And then temporarily, the BCG vaccination increase the immunity against different pathogens. And this is one of the reasons that that we think that it that justify that the disease is not so severe when you are BCG vaccinated, but not when you were vaccinated in your childhood, if you are recently vaccinated.
Thank you very much. We have a question from Mikhailo Ivantsov. What diseases except COVID and TB are you interested and why?
Okay, thank you for your question. Thank you very much for your interest. We have been working basically in the last year in tuberculosis, but we have been also working now in COVID, as a lot of people. But now we are also interested in general in respiratory diseases. Then we have been working in the past with other viruses like metapneumovirus or flu. And now we are interested in patients that are in the hospital that are intubated in the intensive care unit, that the tubes are colonized by Staph aureus or Pseudomonas aeruginosa. And then we are working in different studies exploring how the biofilm it could be disgregated and how the presence of one bacteria it induces the presence or not of the other. And it's not the same if the staph arrives first or if it arrives first the Pseudomonas, and it also there is some difference depending on the strain. And we have been also working in the past, but we also maintain some activity in the detection of pneumococcal pneumonia and also Legionella pneumonia. And we have worked a lot in diagnostic methods for diagnosing the pneumonia for Legionella and pneumococcal pneumonia. And we have also working in the field of mycobacteria, not only tuberculosis, if not non-tuberculous mycobacteria. Then, yeah, a lot of work, but a lot of topics, but in the field of respiratory infections. Thank you very much for the question.
We have one more question from Dmitry Alexandrovich. What do you think, how long COVID will continue and when this pandemic will be ended?
Okay, thank you very much, Dimitri. I had bad news for you. I think the pandemics, well, no, it's not exactly the same. I think the COVID, it will remain forever. The SARS-CoV-2, it will be not eradicated because it's still here. We are not any sterilizing vaccine. Then you are vaccinated and you could be infected in your airway. Then the virus is here for always. But probably the pandemics as a terrible disease over the world, it will go reducing the impact. It will go the vaccination, the mitigation of the attenuation of the virus. I hope it will mitigate the impact in the health. And then in some years, we will live with the virus as other respiratory viruses. And probably when you were the diagnosis of COVID, you will be at home. Yeah, we need to use to this new procedure for some time. Time for the question.
One more question from Daria Zatinatska. What can we do to reduce stigma related to COVID-19, TB, and other communicable diseases?
No, this is a very, thank you very much, Daria. This is a very important question, a very interesting question. And it is something that we also defend because the research teams are always focused in science, medical science, in biological science. But from my view, in this kind of diseases, the social science is key because there is a component in the of the stigma that it should be developed, it should be fighted. Then we, from my view, it's very important in TB and in COVID disease involve as much as we can in our research NGOs. It's very important to involve patient association, and it is very important to disseminate in the media correct information, high quality information about the disease, and to not stigmatize patients. And this is very important to do, very difficult to do, but it's something that it's important. Yeah, thank you very much.
We have one more question. Thank you very much. From Yulia Ishchenko. Can COVID be a new seasonic flu in the future to which we will get used to?
This is the plan. This is the plan. Governments are working, and some scientific societies are working with this idea, because now that we know that in general, healthy individuals with a correct vaccination, it could be infected, but probably asymptomatic or mild, it's the same as the flu. Then has no sense, from my view, to make a lot of effort, all the effort in all population, because these affect a lot the social and economic way of a country. We need focus in COVID, but also in other diseases. In fact, we are doing this in flu. We are protecting the older part of the population, or we are protecting the individuals with some chronic disease. Then we should focus in people with high risk of death or of severe disease. But the rest of the population, she should be cautious, and it should try to prevent as much as we can to be infected. And if we are infected, try not to spread the disease if we know. But yeah, we are going to the flu model for COVID. And I think it could be correct.
Thank you. Dear participants, are you have some more questions to Dr. Dominguez? So I have one. What is special about TB, COVID-19 on TB patient of clinical symptoms or any biochemistry characteristics and so on?
Okay. The characteristics of the TB patients infected with COVID or in general?
Yes,
Sorry.
infected with COVID.
Yeah. Initially, it was expected that patients with the TB patients will suffer a lot with COVID, and the amount of TB patients with severe COVID, it will be high. For the previous lung damage, patients with TB, that is a disease that is long, that you could be ill for 1 year and you could have lesions in your lung for some time. Affected by COVID, it could be severe, it could be worse. But the studies have demonstrated that not always happens, that the TB patients have more or less the same symptoms than the TB patients without COVID. It not seems that COVID in TB patients is worse than COVID in normal population. It depends a lot in the background of the individual, how severe is the TB, of course, and the chronic disease that they have. But it seems that this immune attenuation that a TB patient has at the beginning, it could help to reduce the inflammation with the COVID, and one thing compensate the other. But there is some variation among patients. It's a very good question. Thank you very much, Nico.
Thank you very much. Are we have more questions to Dr. Dominguez?
Yeah, I have one question.
Okay,
do you hear me?
Yeah.
Yeah, I have a question about a lot of vaccines for coronavirus based on spike protein, which are so variable, and for each strain we need to obtain a new vaccine. Yeah, but why medical society don't produce COVID vaccine based on another more stable part of the virus proteins like nucleus or another you understand, yeah?
Yes. Okay, thank you very much. It's a very good question. Thank you very much. I think there is a multiple reasons that justify this, but also multiple reasons to justify that it's better to combine. On one hand, I think it's clear that the spike is the more immunogenic. Then it's more easy to perform one vaccine against using only one antigen. That in addition is easy to produce, and it is producing a high immuno response. I want to say now that we are lucky with the COVID in some way, because the immune response is very easy. The structure of the virus is very easy, and it was very similar to the MERS and the SARS-CoV-2. Then we have a lot of work done, and with one antigen, you are able to generate a very good protection. Of course, the idea is to combine new antigens in the future and to update the vaccine because the new variants are changing. And then, from my view, the combination of vaccines that are using different antigens is a good thing, because you are stimulating different subtype of T cells, and then you are increasing your protection. In my view, there is room for different kind of vaccines. We will see what happens with the new doses for everybody in the future, if it is coming or not, or it will focus only in individuals with high risk. But the real thing, or the real need that we have, is a vaccine that avoids the infection. Because if you are infected and you could transmit the disease, we will never stop the transmission. We will never stop new contagions. That is very good if you are infected and you are healthy, of course. Then probably new vaccines that are administered in the nasal mucosa, it could be good, because it could stimulate the mucosal immunity, and maybe we will reduce the risk of asymptomatic individuals. Then this will be good. Then in the world of the vaccines, a lot of work to do with a good idea to combine different vaccines for sure, because it increases the different subtype of T cells responding. This is a good plan. Thank you.
Thank you. We have one more question from Mikhail Ivantsov. What COVID vaccine is the best now to your mind, and will there be some new ones in the nearest future?
Okay, I have answered a bit with the previous one. I think all of them are very similar. Probably at the beginning, the vaccines based in RNA, messenger RNA, was a bit better, but now with the adjustment of doses, and yeah, I think all of them that are available are getting protection that it could help a lot in controlling the disease. And in the future, we will see probably combination of them, and probably more focus in risk patients, high risk patients.
Thank you. And one more question from Daria Zutinska. What should TB programmers do to lessen the impact of the COVID-19 pandemic on TB prevention and care services?
I think the TB program should use a lot of things that has been developed for COVID. And one thing we can know every day the amount of TB cases, the COVID cases, every day. Every week you could know how many vaccines, how many persons has died, how many people has COVID. And for tuberculosis, we need 1 year. This year have the data from the last year. And this is something that it could be changed. If we are able to know for COVID, we could do also for TB. We need also to learn about the diagnostic of COVID. It has been developed a large amount of very easy to perform methods, antigen and molecular. Then this invest, it should be also performed for COVID, to be able to diagnose the disease in rural areas, far from the big hospitals, to reduce the burden. Yeah, and the third thing probably is to make an effort to reduce the inequity, because it seems that the COVID has received high invest in a very low time, because it's affecting some countries, and tuberculosis that is affecting others, it don't receive the same amount. And we have worked a lot on this.
Okay, thank you very much. Any additional question to Dr. José Domínguez? Okay, if not, I want to say a very big thank you for your very interesting and complex and very important actually lecture, because yes, we see that we have new problems like COVID pandemic, but old problems don't get somewhere, and we should, I think, build some opportunities, some approaches to control, and don't waste the time to control this terrible disease such as TB. So it's this world initiatives like Innova for TB and other projects is very important, and I'm very proud that Odessa National University, in collaboration with Dr. Dominguez and some other scientists, took part in this world initiatives. Thank you very much, Doctor, for your collaboration with us and for very, very interesting lectures on our summer schools.
Okay, thank you very much to you. Thank you very much for all the attenders, for the very interesting questions. And it's a pleasure for me always to participate in this very, very nice summer school. I hope to see you soon.